seed production, especially maize, is isolation. You have to get good isolation, and if the varieties are plenty, then it becomes very difficult for you to get the right isolation for seed production. The way forward, we have some of the seeds in the cold room. Now, I'm very confident that this year we get buys, because last year the weather was bad, so there were poor yields, and it looks like most people did not have or do not have adequate quantity of seeds. So it's most likely this year all the seeds will go. And then the Cassava Gcipp. This was only for 9 months, but 9 months. It also ended at the beginning of the year. And the objective is the same, to produce cassava planting materials for in support of the government planting for food and jobs, and also support the government policy of one district, one factory. And we have partners. The partners, the AGRA contracted us, contracted us to produce for a certain partners. And we did 2 plantings, one at Arowa and one at Amantin. The Arowa one has been distributed and is finished. The Amantin one, 35 acres, and we have it currently on the field. We planted 35 acres last year, and these are the varieties that were planted. These are the popular varieties that the farmers require. We did the planting last year, and then we did all the activities with control. AGRA people came to inspect the field, and they took pictures. And the 35 acres that we had, we did some distributions. So Josma took 20 acres of them. Hompong also took 20 acres. KNUST Agri also obtained 7 acres. And the Mountain Agro, because they gave us the land, they took the bulk of the planting materials for more than 100 acres. And then others took about 10 acres. But the good news is that because the funding for this project has ended, they have re-established the 35 acres for us, and it's for the institute, and at their own cost, because they didn't buy the planting materials earlier that we gave them, more than 100 acres. So they are compensating the institute for the 35 acres at their own expense. So the undermarked activities. Now I'm reporting for the other members of the marked seed production activities. So these are the members who are involved. Now initially we were to produce maize, rice, soybean, cassava, and pepper. But this year we didn't produce pepper. So the locations were Fumesa, Sokwai, Kwadaso, Yabe, Ohau, and Tanos. Now these are the seeds that we obtained. They set target for us. And for the hybrids, we produced Ahofe. The target was 0.326 tons, and that was achieved. And then for CSIR Nkomo, the target was also 0.326, and we achieved only 2.60 because of adverse rainfall. Now for rice, they achieved their target. The target was 1.5 tons, and it was achieved. And they were to produce 2 varieties, Amankwatia and then Agra rice. For cassava, they also have under Agra 3.5 hectares, and the various varieties are indicated. For sweet potato, they were to produce 28,000 vines, but they managed to produce 10,000 also because of adverse weather during the minor season. Like I'm saying, the interseasonal drought was a big challenge last year, especially in the minor season. In isolation distances for hybrid parental maize seed production is an issue. Way forward, the activities will be continued this year if funding is made available, and we'll collaborate with MOFA and Grains Board. These are the publications I had last year. Conference abstracts, 1 training manual, 1 referee book chapter, and then some media engagement. Thank you very much.
Great. See, Dr. Allen, you were on time. Because you were my immediate past head of division, I was afraid to tell you to stop. You see. You will agree with me that we've had very nice presentations. We now invite the participants, the presenters, for interactions with them. Great. I have a few general remarks to make, then I hand over to the director and the senior citizens, and then to the general house. I have questions in fact, no questions, just to remind you of certain things, to 2-3 people, and then I give general impression. Esther, I was seeing bacteria wilt, but I think the bacteria is the plural form of the pathogen bacterium, but if you are talking about the effect, it is bacterial. So we have bacterial wilt, not bacterial wilt. So we check that. Fenting, I saw a scale of 1 to 9. Has it been developed already, or you are now trying to put that together? The last one is 38.1 to 60 for the 9th scale. Are you developing it, or you are? So keep that one time counts for. I think the presentations were quite good. Particularly the slides were very clear, and our presentations good enough. Now my concern is that those who presented tables, in pathology, plant health, we tend to meet discrete data like indices and populations of insects, nematodes, colonies, and things like that. Such data must always be what? Transformed. Because if you don't, with some publications, they will not be accepted, some journals. So when you do the transformation, you put that in brackets. It is the transformed data that you use for the analysis. But for reporting, you go back to the raw data. So please take note. The other thing is, I was happy that those who presented graphs had the error bars. That is very important, because they are indicative of the variances amongst treatments. Once again, some journals will not accept if you draw the raw this thing without the bars. So it is a very good thing you did. Then also the other good thing is that virtually all of you were collaborating with other divisions. That must be continued, because the pathology work is on crops, mandate crops, and there are other staff members working on these crops, particularly the breeders. So always we have to have them in their in our groups. The last thing is to Dr. Dansu. Work on the division. I was expecting to find something on director because he is part of your division. That is important. When you were given the research work of the various scientists, you see, then Professor Lamtitu, you indicate his area as at now, they mean the college, and then myself too. You see, and Then, so you know, I have a very small project, you are aware. For your information, yesterday they said that the one will be coming anytime soon. But because you never mentioned it, you will not chop some. Okay, you're not going to chop some. You know, it's a project, student project away from Benin. And they've asked us to do a bit of it. So Dansu knows it is small, so he will not talk about that. And the punishment is that he's not going to chop anything from that. Director. No, not the player. The student.
Okay, thank you very much. I think I agree with you that the presenters have done very well. And I also share most of the concerns raised by Dr. Osei. Now to the specifics. Dr. Danso, I followed with keen interest on the work that you did using the two treatments, super attacker, and then insect pathogenic nematodes. I think the super attacker recorded mortality of 100, you said that, and that of the insect pathogenic nematode 79. But I didn't see the significance between the two, the 100% and the 79. So what was your conclusion? And then you were also talking about using the third stage of a larvae or so in an experiment. And I want to know how you'll be able to use that and not any of the insects, 2, 3, or 4. And Dr. Opon, I want to commend you for doing most of your trials at our outstations. I think I saw about 3 or 4 trials being done at our outstations. I encourage you and others to send some of the trials to the outstations. Dr. Amwabeng, can you tell me the key findings of your work that you did on cabbage, especially those from the districts that did very well? There were no key findings or major findings. And Blanson and then Mr. Sander, you are trying to explore the use of biological candidates or biological control agents. And you know that you agree with me that there is indiscriminate use of these insecticides. How sure are you that you will succeed? I want you to comment on that. And Dr. Enin, I also got excited about your cabbage trial, but you were using a lot of insecticides. For example, I noticed that you said that your spraying regimes, you used spraying bi-weekly, and you switch on to weekly spraying of these insecticides. But me and you, or you and I, know that we normally prefer monitor spraying as against calendar spraying. I'm tempted to believe that you were using calendar spraying, since it was bi-weekly and then we switch on to weekly spraying. And I think Dr. Ose has commented on that for Mr. Fenton, the disease score based on the scale of 1 to 9. I think if it is not you, you need to give credit to the person who developed that protocol you are using. And then Esther, the first graph that you presented on incidence and severity, I think I didn't see any labels for the Y and X axis. So take note.
Thank you very much, Mr. Chair, and good presentations. To Dr. Danso, um, you said you issue import permits. I know it used to. It is still ongoing, and if it is, what clearance do you have from PPRHD to do that? Then the nematode you are using for the control of fall avenue, what are the economic benefits of it, of using the nematode? If I got your concept right. In fact, all of your presentations were good. I was wondering the font type and the sizes you used. It was very legible. And I'm wondering, as an HOD, do you think you can set up a consortium to consult for commercial farmers on pathology issues, disease-associated issues, so that we can use it as an income generating slot for the institute? And Blackson, please avoid crowded slides. Some of your things, I felt they could have. I don't think we are restricted as the number of slides we can have in a presentation. And Uma, I'm wondering, these days the rains, the weather pattern is changing. How is that going to affect your sample collection that you said it usually happens from July onwards? How is it going to affect it? And for all of you, those testing, identifying pathogens, bacteria, insects, whatever, what molecular tools are you using to enrich the work that you are doing? Esther, your presentation was good. You spoke to us most of the time. And again, the incidence and severity scale graph that you showed, I think you could have used both sides of the y-axis, one for incidence and one for severity, so that one wouldn't be hiding at the bottom. Then the CMV, you are talking of advising the cassava mosaic virus. You said it will. Your conclusion is it advises farmers, the breeders. How about farmers, and how about the concept of planting material dissemination? Then the bacterium wilt. What advice would you give to breeders with that? Apart from breeding, what other control measures? You said it's a guide for the breeders to select. Apart from that, what other system would we use to control the bacterium wilt if we observe it in our fields? Dr. Upon, good effort. And how about what is your take on the use of clean planting material to control the viral disease pressure in the country? You were saying you are looking at isolation sites for seed, maize seed. Asimfosu, I know they are asking for breeders to come around. They have fine areas that you can do your isolation. And what do you think would be do you think it would be very beneficial for us to maintain the Amantin and our sites of cassava? You know, I don't know what advises the cost of cassava planting material, but the money you get out of it is, I think, the copicing fee. So would it be cost beneficial for the institute to maintain it? Thank you.
Thank you very much. I also want to commend all the presenters. The slides were very catchy. I would like to find out from Dr. Yadanso, why did you use only Adonten in this your insect pathogen nematode function? At least if you had used about 2 or 3 varieties.
And then why did you select Abontem? What is special about Abontem? And Dr. Allen, you've been working on aflatoxin maize for quite some time. How soon are we going to release the first aflatoxin tolerant maize in Ghana? And then Dr. Frimpong, farmers ranked the farmers ranking. How did they do it? And how did you correlate it with yours? Dr. Joseph Adumaku, you identified root knot nematodes in common beans. Do you have any options for managing root knot nematodes in common beans? And then Umar Sanda, you were talking about the challenges in acquiring commercial pheromones. How are you going to address this challenge? And then Esther, positive selection has been going on for quite some time. What are farmers adopting? So I want an idea of whether farmers are adopting or not. Thank you.
Okay. Dr. Yaudanso, when you started your presentation on the division, I saw you confusing section with units. Though you have units there, then you're talking about mycology section, biology section. Plant health has 3 sections. In your presentation, make it clear. Plant pathology, weed science, biocontrol. Entomology and biocontrol. These are the sections. And then the units are the mycology, biology, and all those. Okay. The import permits, I think the deputy, I was also concerned because I knew it was an issue between us and PPRSD. So please enlighten us as to why you are still issuing import permits. Now, I want us to. Well, I don't know whether they even still doing it or what. I want to be clear about that. Yeah. Then I want you to tell us, as the head of the division, any success story about the management of fall army well. I think some work has gone on. Is there any success story? Not on in a lab basis, so whether farmers have been adopted any management strategy. Is there something you can tell us? I don't know. Then, Mr. Blanson, I saw something, a research team. Then you were listing names, and then I saw ETA. I don't think it's too appropriate. I mean, this is not citation. Research team, if it's technical staff, right? Technical staff, ETA is not appropriate. So next time, don't do that. Dr. Adomako, you saw the common bean. You saw some plant, showing chlorosis, and you went further and you found out it were nematodes, root knot. Did you identify any other pathogen apart from those that have chlorosis? Any other pathogen in the field about the common beans? I'm interested in that. And I want to find out the disease situation in other parts. This will be seen in your field here, but in other parts of the country, the common bean, have you been identify any diseases? I want to find out. And then, do you have plan to screen the materials here? Those that you really find out that nematodes, root knot nematode was very, very important on them in other locations for us to confirm the importance, because this is the first time I think you are seeing these things. They are about the root knot thing. So that at least we will be able to confirm the root knot is maybe even the major pathogen on common bean or whatever it is. Do you have plans to also screen in other locations? I want to find out from you. And then your work on the bacteria wilt and the nematode interaction. Any yield losses about the interaction? I mean, the Rastonia and the nematode interaction. But what are the yield losses? I want to find out the importance of that interaction on the tomato. Yeah. So tell us whether there are any recorded yield losses. Fenton, Jerry, I'm becoming uncomfortable about the way you are describing skills. I don't know whether it is from where your reference. Because I find it difficult to understand that when a scale of 1 to 9, we have a score of 3, and you say it is resistant. I personally find it difficult because fundamentally, if a plant is resistant, it means it's not showing any symptoms, then it is resistant. If it is showing some mild symptoms, it could be highly tolerant or so. Then we go on, moderately tolerant. But when and the same thing with even Esther, some aspect of your work, let us be clear on that. Please, if it is resistant, then it is 1. If it is 3, then let us find an appropriate thing to describe it. I'm becoming very uncomfortable with that kind of description. Esther, for the sake of getting a publication and high impact journal, I'm talking from experience, I will be glad that you confirm your ELISA results with an RT-PCR test. So that at least that one, you can be sure you can publish your paper in high impact journal, because I know some journals I'm talking from experience. When you stop at ELISA, they have problems. So that is my advice. Okay. So for now, I think that's what I have for the plant.
I think they already have had enough. We shall come later on. And if we should add some more, they are reacting to this first. Because if we should add more, some of my people might collapse over here. The questions are just too many. So,
right, thank you. I think the first one has to do with from the writer about the spraying regime. In fact, the farmers, they spray every 3 days. And based on our projection, looking at the life cycle, the developmental stages, we thought it that by starting with 2 weeks, we should be able to estimate when to spray. And in fact, before we do the spraying, we assess the population. But one constraint was that if we were to use the threshold strategy, that means we have to be going there frequently, which the resources could not permit us. So we used that, and we were also communicating with the farmer to update us regularly on the pest infestation status. So that's why we use what I would say a merge of the calendar spraying and the threshold strategy. And Prof. Hans, about the ranking, I think has gone out. Okay, about the ranking, but we use the number of plants infested and the damage level as has been described for assessing damage by the diamond bark moth on our part. And we also allow the farmers to visit all the plots. And though we had 10 insecticides, in order not to confuse them, we asked them to rank the first 3 in that order. So the farmers, based on the damage levels and the pest infestation levels, they rank those performance. And also, I think my divisional head presented something on the import duty and certification. I think I will step in here. I think previously the regulation was lax, let me put it that way, and that we had the leeway to undertake such activities. But of recent years, I think the mandates has been given to PPRSD, and based on their acts and also their guidelines, we even have to send our samples to them for certification before we send. So it's a part of our activities. But for now, because of certain laws that are coming up, we are not able to undertake that activities. Thank you.
Thank you. Prof. Nancy, apart from the nematode, we also identified the angle leaf spots. There are viruses, or let's say viral diseases. We saw the other leaf spots and fusarium mold. In fact, from this work that Jerry also presented, the continuation of some of the fungal pathogens we isolated. So it's not only the root node nematode. But what my interest was that normally we neglect. Once we see the chlorosis and that thing, we might think it's fungal infection. And that was what I went in, because that was what literature and other things were saying. But, like I said, because I'm skilled to nematodes, I also decided to go further to find out what was really causing it. Then the screen was done in the survey was done in Ejura, Akumadan, Amantin, and Fumeswa here. That's across our research fields where they were having the common thing. And it was across the whole field. So indicating that the nematode is now becoming a major pathogen we have to take care of. And the bacteria and nematode interaction. Yeah, bacteria alone are reported to cause major loss, about 90% or 80%. It becomes more devastating when the nematode attack also. Then it means that the plant will have to now fight two enemies. Nematode is causing its own thing. The bacteria is also causing its own thing. But the point is that the bacteria becomes more aggressive or more deadly because the nematode is giving this a wound for it to also attack more. So in other countries, they've reported of complete field loss where these two interact. But in Ghana, you have not come across any. But I've come across bacterial works, but not the interaction of the two. That's why I want to go further to see how, if these two come together, what will be the level of devastation that we're going to identify on our fields. So that's what we are now going to do and find out if these two come together, what will be the extent of the damage that we are going to experience. But in most countries, they've reported complete full loss and other things due to these interactions. Then Prof. Hans asks of the management of nematode. I think the Plant Health Division have developed so many technologies. Each year reports, but we'll go for, until we identify a resistant material, I would advise that we take very good care of our food sanitations and cultural practice. Most of these pathogens thrive very well and best because we don't keep good food sanitations. The way we handle our fields may not always be the best. So if you are working or you're advising farmers, so like Deputy said, maybe the pathology, the plant health can form a consortium and be advising our breeders, IGF, and charge them and other farmers. So please, when you are doing your food, it doesn't cost much to get somebody from plant health monitoring your food or advising you on what to do. But please, let's always maintain the food sanitation is very important. Thank you.
Dr. Say, and then Director, your comments are well noted. The others, I will look at it, and then next time I will leave it. Thank you very much. Deputy, about advice to farmers on the cassava mosaic virus disease. I would advise that the farmers should use improved or resistant varieties. They should purchase it. They can also fall on tissue culture materials. I know that the Institute work on cassava tissue culture materials, which it is available. And then also they can also practice this positive selection, whereby they would not select those that are highly infected for planting in the subsequent growing season. So that's my advice for farmers to control this cassava mosaic virus diseases. And then for the bacterial roots control, we can use rotation cropping to control it. It's a soil-borne disease. The bacteria is found in the soil. So once it's there and the farmer identifies it, the best thing is that you uproot those. If they are not many, you have to uproot it and then destroy it. Then you plant in the subsequent year, you plant a non-host crop. You can plant maize, which will not, which the bacteria will not attack, so that by the time you come back to your tomatoes, the soil would be somehow clean for to prevent the incidence of the diseases. And then for Prof. Hans, adoption of positive selection. Yes, positive selection. This was under the Gcipp project. Adoption was very high. That is what Dr. Jonas used for his work, and he worked on adoption. Farmers were ready to adopt, but I think the post effect of the project, I don't know, but I think during the time of the project, farmers were so much interested to learn this technique because it's easy. It is easy to identify a disease or an infected plant and then just isolate it or take it out from the lot that you have. And so farmers are willing to adopt that. And then Prof. Lante, yes, confirmation with RT-PCR, it's much welcome. I will consider that. Thank you very much.
Thank you very much. Dr. Hussein, your comment is well noted. I know you are key members of the division, yourself, Prof. Lamte, and Dr. Grace Bofriaku, as well as our director. Sorry for the omission. Director, you asked about the conclusion on the performance of the super attacker and that of the nematodes on the knockdown effect. The structure of the experiment was that we built a bigger plant house and partitioned it. So each compartment accommodated one treatment. So it is a percentage of the number of larvae that were applied. So for each cage, it wouldn't have been possible to be replicated, to be analyzed statistically. So 24 hours after the treatment application, we went in and collected the larvae that were dead and put a percentage on it. So that was the structure of the experiment. We didn't have enough funds to have constructed several of the cages to. Thank you very much. The second question was that why did we use the third instar stage of the 4-lammy worm? The work was done in conjunction with the entomology session. So they gave that advice that stage, either second or third, it is the most destructive stage of the insect, where its destructive activities will be seen very much. Prof. Marian, economic benefits of insect pathogenic nematodes. Insect pathogenic nematode work started barely 3 years ago at the institute, and we are recording promising results. So going forward, we hope to carry the experiments to the field, so that this economic analysis thing will be done. But basically, in other jurisdictions, they are called economic nematodes. They are commercially sold in agri-input stores. Farmers go and buy them and apply to kill insects harming their food crops. So this is a new area for us. We are still studying the system. Very soon, we are going to do full studies, and those other things will follow. Prof. Hans, you asked why did we use maintain maize variety. This that particular work started in the year 2019 under Dr. Salah Amaini and Alaji. When the work started, the funds came in very late. So we wanted a variety that was extra early or that could yield fast and give us results. So that is why we settled on that particular variety. It is extra early variety. Any success story on fall armyworm management in our division when the I would say that so far so good. If the nematodes work and that of the